So it's really a pleasure to report here today. The talk will be around 40 minutes, and feel free to interrupt in between for any questions. So first, a very quick summarization of scGPT, also a quick intro for single-cell foundation models for people who are not familiar with this direction. This will be followed by my personal takes on several lessons learned after the release of our work. What are single-cell omics and why we care about building foundation models for it? Different from bulk level readout, single-cell sequencing provides cellular scale resolution and reviews cell heterogeneities. Recent advancements enable multimodal sequencing across the central dogma. For example, a TAC-seq can be used to measure chromatin accessibility, and SAT-seq measures RNA expression and protein altogether. And recently, CRISPR-based perturbations have also been combined with RNA sequencing in the perturb-seq technology, so that effects of genetic perturbations can be recorded now in high throughput. This whole reservoir of technologies can provide comprehensive view of individual cells. So today we will focus on technologies mostly read cellular RNAs, for example, the single-cell RNA-seq. And the reason for that is that the RNA-seq comes in larger abundance. You may find around more than a few hundred million varying cells in public domain, and the technology around RNA sequencing is also more mature, meaning that the data quality is relatively higher than other modalities. So these features make it suitable for training data. So make it suitable as a training data for large language model to answer relevant biological questions. So what are the biological questions we care about? Once we have a foundation model, or even nowadays people talk about virtual cell models, we can use it to answer questions from more biological related ones like cell type assignment, gene regulatory prediction, to more therapeutic related questions such as predicting perturbational response, or the predicting the differentiation trajectories of your different cell products. More importantly, if we think about it, these applications are interconnected intrinsically. For example, as visualized here, solving gene interactions can greatly contribute to the solution of other questions, like how cells differentiate. So these connections justify the rationale of foundation models, as it can learn from and provide a shared biological context across applications. Now, how did we build a single-cell foundation model? We first care about data and model architecture choice. Well, texts are made up of words. Cells can be characterized by genes. This inspired us to believe that transformers would also be suitable to learn interactions between gene tokens. And the second is that we already have a vast scale of sequencing data, such as shown in this screenshot from the cell by gene census, which contain tens of millions of sequence cells. This screenshot here was actually from 2023. And if you look at this, a public data corpus really grew fast in accelerating speed over the last few years. We have 33 million cells in 2023, and now in the same corpus, there are 108 million cells in 2025. Therefore, one can train a large model via self-supervised learning to build a pre-trained foundation model that understands cell states. We conducted an autoregressive generative pre-training. In the pre-training, large-scale expression matrices were collected and processed. We selected around 33 million human cells of 51 organs and tissues from the Chan-Zuckerberg cell by gene collection. And our model differs from transformers in the natural language domain in terms of token embeddings and the transformer model itself. For the input embedding, we use the 3 sets of tokens to represent the single-cell RNA-seq data. First, it's embedding for gene identifiers, which map all the gene names to the vocabulary of learnable vectors. Second, we use a fully connected neural network to encode expression value into embeddings. And also, I want to highlight in the pre-processing stage, we introduce the value binning technique to normalize the data values across studies. Third, we include a flexible condition token. These are flags of possible metadata, such as whether the gene expression is known or to be predicted by the model, or later on, if you fine-tune the model, this flag can represent whether this is a gene being perturbed or it's not. The core model itself is a 12-stack transformer blocks, which customized mask attentions of generative pretraining. Briefly speaking, the model is trained to reconstruct unseen genes, given the mask extension to guarantee only the relevant information of already seen genes can have attention. The output of the model is this HL, which is a group of vectors with the length equaling the number of genes. We also reserved a special token called the CLS token at the beginning. So the output had this CLS token, at this position of the CLS token can represent the integrated information of the whole sequence. In other words, it represents the cell embedding. So scGPT supports varying applications after the pre-training. I just want to quickly highlight some of them in one slide. These tasks are achieved either by fine-tuning the model or zero-shot calling of the model itself. I especially wanted to highlight one of the tasks here. We built a fast reference mapping using the cell embeddings. So one can map and annotate cells by querying similar cells from large cell atlas. Since we can store the atlas embeddings, we only need to compute the cell embeddings over millions of cells in the atlas once and then store them. And every time we have new cells to query, we just encode the new cells and also do the very fast nearest neighbor search enabled by other very efficient packages. And by doing this way, this reference mapping can be conducted very fast on GPU within seconds. Here in this screenshot, I show that I think this for around 4,000 queries. You can finish that in just about 0.1 second.
I have a quick question on that one. So originally, scGPT was not trained with any type of metric learning loss. And in this one, basically you are querying the closest neighbors to a given cell and using that to annotate it. Is that correct? And in that case, what does it ensure that our nearest neighbor queries are like meaningful in this space?
I think you talk about query by the annotation.
No. In this case, you are basically getting the distances of your new data set versus your atlas, right? So you have
Yeah.
your atlas cells embedded and you have additional cells embedded, and then you're trying to do a k-nearest neighbor search
Yeah.
to find the closest. And that relies on, and again, S similarity was one of the few that actually introduced a metric loss because they wanted to do distance-based queries. But I was
Yeah.
wondering in this case, why do we think that the metric property of like embedding being meaningful for distance-based queries hold?
Oh yeah, so I think I have two steps of my answer. The first is that because even before introducing this task, after pre-training, we actually look at the cell embeddings. Just plotted on UMAP, you can see that the overall the high-level structure does really correspond to cell types, or the first level of structure or similarity between the embeddings are really biological related features or distance like cell types. So that's why we think you can use that to encode new cells. And one of the second part of my answer is that because I think why this works for a pre-trained model but not for other models before is that actually in the pre-training we already pre-train on many hundreds of data sets from hundreds of different studies. So in a way, that model already see different batch effects. We have some additional technique to partially remove some batch effects, but overall, it's because of this large training. And now you have a embedding system that can encode different data sets, meaning that if you include just one more data set, it's fine for the space. Yeah.
Yeah. And we also have a very related question right now, which is exactly talking about the similarity which I just brought up. if you have had a comparison with a similarity, either in terms of speed or sensitivity for retrieving cells, because that's again one of the methods that explicitly trained with the goal of being able to do distance-based queries. Have you had any chance to compare scGPT performance in the nearest neighbor queries versus the a similarity that was released?
Oh, I personally didn't do that, and I think there might be one or two benchmarking papers that actually did that, but sorry, I think I forgot the exact title for the paper, but I'm
Yeah.
willing to ask more, where such a. Yeah, but generally speaking, I will actually discuss more about this. I think it does very well for core cell types, but not very well if you look very carefully for, subtypes or,
Yeah.
very low types. I would actually discuss that in later slides.
Yeah.
Yeah,
Thank you.
yeah, no problem. So apart from that, another interesting task we did during the fine-tuning stage is actually perturbational response prediction. So this task is that the model learns to predict unseen gene perturbations result from known experiments. You learn from some of the observed ones and try to predict unseen ones. To predict the genetic perturbation response, the model we train such way and tested on in our original paper, we tested on 3 different data sets, and we compared the performance with other state-of-the-art model at that time, which is like the Gears model, and we showed that scGPT really generate a very nice result. And we also visualize some of the cherry-picked examples for specific genes. You can see the predicted, so the blue ones are predicted. Sorry, the red ones are predicted. The predicted ones actually match well with the distribution of the genes expression in the measured ground truth. So we also trained a reserve perturbation task, which is a opposite way for the previous slide. Given the resulting cell state, we want the model to predict the source perturbation. This was showcased on the rather small subset of the data, which only includes 7 test cases. On the left, we show that the most successful cases, where the true perturbation combination is retrieved within the top 1 or top 2 guesses. Overall on the 7 test cases, we saw a higher hit rate is observed among is observed when compared to Gears as a baseline or another baseline of calling the most differentially expressed genes. To summarize, scGPT provides a general pre-training workflow for single-cell data and supports varying tasks. We were fortunate that scGPT becomes a quite popular tool in the field, and it gives us a chance to communicate, learn lessons from our users. The first lesson is that we observed and found that scGPT indeed can generate biological meaningful cell and gene embeddings. Here I show an example of a very large retina atlas collected by a user group. You can see the pre-trained embedding align very well with the human annotations generated by the experts. When they tested on the newly collected data, they usually see, whenever users test on similar data like this one, they usually see a over 90% accuracy of this core cell types, and usually they can be quite happy about this. This is a quite common case we heard from users, but what our users are usually not happy about is that when looking at more subtle cell states and subtypes of cells, this is what remains being missing. For example, actually this one, this very large blob of row cells can contain over 100 annotated subtypes. The pre-trained model does little job distinguishing them, at least not at this higher level. Now for this case, we actually collaborated with the users and later on annotated these cell types very well with fine-tuning of the model. However, I do want to recognize, this missing piece, this missing capability of the model. We surely want the model, and surely it can make more sense to have such ability right after the pre-training. We also observed initial success about perturbation predictions and driving gene identifications. Here, as I mentioned in previous slides, here I also borrowed a figure from the GeneFormer paper to show that the success pattern about in silico perturbing and reprogramming the iPS cells. However, the hit rate can be still quite low in real life when users actually try, applying the model on their very large perturbational data set. We need more reliable predictions, especially in the pre-training model of the shell once again. So one may just suggest that maybe scaling the model is the very easy solution for this. Well, we did find some scaling up we did actually find that scaling up may not be a simple solution as one may imagine. In a word, a universal scaling law is still not clear for single cell models. So for ourselves, we reported very early a scaling effect in our paper, particularly regarding the data size. So a larger pre-training data size yields a superior pre-training embeddings, and then that further lead to improved performance on downstream tasks. So this one is shown on the cell type annotation tasks. But however, if you look at it carefully, you see that models, the performance reaches a plateau after around 3 million cells in the pre-training, which is actually only around 10% of the training data in the our scGPT training. And we actually have seen this kind of phenomena even after the release of our model. On the similar topic, actually related to single cell, foundation models, we also saw the mixed result reported in protein language models as well. I want to borrow this example here from the model of ProGen3, which is a protein language model. And on the right-hand side, the figure shows that the fitness score on one of their evaluation tasks actually did not improve after the models, for models beyond 3 million parameters, the performance did not improve. But if you look at a pre-training, you see a different story. The pre-training probability actually does scales very well for the ProGen3 model. This actually, I use the example to give me a hint to show a possible answer for what's actually happening here is a mismatch between the tasks, between the training tasks. So unlike in natural language domain, where is the ability to generate the next tokens in GPT-5, the GPT-5 is trained in that way to generate next tokens and actually using that way to generate sentences. But for protein language models and for single cell foundation models, we face this kind of challenge in the same way. The pre-training is usually on reconstructing cell states or reconstructing the missing piece in the protein sequences, but the model later on is used in other ways. So naturally, we envision a future improvement for this is to have aligned pre-trained tasks, which I think that may potentially solve this scaling law issue. We will discuss this in later slides. And to envision the future of how to train a better foundation model, let's look at the ongoing trend of the multimodal multitask retraining I actually see in the last year. So after the release of scGPT and of GeneFormer and UCE, we see a lot of new progress in the last year. They can be largely characterized into 5 directions. This is actually a personal thinking. So including multi-omics, dynamics modeling, including spatial omics, and also efforts for integrating human knowledge into the pre-training itself. And very lastly, but also important, about scaling up the model in a more scale up capable way. So also in the field, we saw a diverse field of new pre-training tasks that can be added to the pre-training stage and fine-tuning stage as well. Collaborators and I also personally work on some of these directions. For example, we recently released scGPT Spatial and also the MesoGPT for epigenomics modeling. Here's a quick overview of scGPT Spatial. It is an extension of scGPT onto the spatial omics. We collected around 30 million data points for spatial data points and also continually pre-train the model on this spatial data. So again, it can be used to address many applications of spatial data analysis, such as annotating the interesting regions in slides and impute unseen genes or cell types due to the limited resolution of spatial profiling. Although this high diversity of progress, it does make one wonder what is the most relevant next step towards a virtual cell model, a much more capable model. For me, I think the answer is dynamics modeling, which is a better way to use just a name I give to summarize the direction about perturbational pre-training and also about training the developmental states of cells. So the reasons are twofold. First, dynamics modeling is an example again for this direction that is highly relevant to therapeutics discovery. So it is quite important. You can imagine we can query the model in a way to ask about what are the genes to perturb, and then for target cell state. Imagine this can be your iPS cells, and this can be your cell product. You can be really interested in silico screening that can give you some genes of interest to control the cell differentiation. And another perspective is that we can say that this new dimension, the dynamics pre-training or predicting the cell dynamics actually opens this new dimension. So so far, most of the cells are trained on, most of the models are trained on static single cell data. Although we have different applications, different tasks, it's like we are expanding on this single y-axis, but opening another axis actually can give you a much more application. Once with a uniform, we envision in this way that for pre-training the model in a to capable to predict dynamics, we can actually put the model once again in a unified tokenization and also unified tasks. In a recent perspective work, we discussed this promising direction of training unified transformers, where you can have hybrid multi-level of tensions to enable the promising processing of tokens of different biological layers. And you can have tensions across different modalities, across different biological layers to learn the interactions. And this can be a unified way to include multi-omics data, and also a unified way to include multitask in your pre-training. So this is an example of a visualization of how we can unify all the pre-training in like a sequence-to-sequence way, but you can give the model different prompt tokens, like task tokens and meta tokens, to control what are the predictions the model is going to make and going to be optimized. The task we already have is like this one, like gene expression reconstruction, but we can also prompt the model to do temporal predictions, or more relevant to us is perturbational predictions. You can imagine you're giving the model the current expression of the cells or the control state, the profile of the control state, and then try to prompt the model to predict the expression of the post-perturbational state. And to having all the different training tasks in a unified way, we envision that this can be a good strategy to expand the current pre-training and to have more capable to integrate more ability of the model in the pre-training stage. But actually, with this being discussed, I actually think the model arc and the training task itself are more straightforward in terms of a straightforward part of efforts we can make for this direction. The more interesting part can be, and more important one can be how we can build a model that can co-evolve with the data, with the data itself. Now, to illustrate this idea, let me introduce a recent work of us in a related but different field of building a model-based system for drug delivery. So this is especially about finding new lipid nanoparticles for delivering mRNA drugs into a specific tissue. So what we're showing here is that we have a self-driving lab system, which we call LUMI Lab. And here we have 3 robots actually in the system containing all consecutive steps from synthesizing the lipid nanoparticle, the LNP, and then delivering the LNP into cell, and then also test that in vitro and a readout and feedback to our in-house chemical foundation model. So this is really a closed loop that on the front end, the robotics generate the data. And then on the back end, we have a chemical foundation model we built that can propose new LNPs, and then that will be tested again in the next round. We actually achieve a pretty high throughput of 100 new LNPs per day per machine. And we use that to in iterations to find a new powerful LNP system for delivery. Here is a quick video demo, 1-minute demo for them. So I don't think the music is playing, so let me briefly introduce that. So you can see the system working in progress. This includes 2 liquid handler steps. You synthesize an LNPs and also delivering the LNPs into the cell in the second one. The next part is that you can have all the system on board, including some of the figures are showing the cell plate readers and also the cell incubators. You record the system in each iteration, record a readout of the system in each iteration, and then the system can automatically propose the candidates for next iteration and start the next iteration. So using the system, the LUMI Lab actually contains 3 stages of training. This is about the chemical foundation model in the LUMI Lab. The first stage, we have a 30-minute generic molecules and their 3D confirmation of that. We trained a model on this generic molecules for the first stage, and then next to them, we have a continual pre-train on 15 million more lipid-like molecules. Things the application of this self-driving lab is about finding lipid nanoparticles. And the third step is about active learning with the self-driving lab iterations we just showed in the previous video. We did find the stage 1 and stage 2 pre-training does show improve the performance if we compare the different modal checkpoint in a simulation. And this is on the simulation of around 2,000 data points. And the model is compared with some other works like GNN network, which this is a more clear, a pre-trained GNN pre-trained graph neural network, and also compare with a molecular descriptor model as well. And the whole process is backend. I want to emphasize this is part of why I think the data and actually the model design should actually collaborate together. So we deliberately designed a chemical space, a large chemical space of 220,000 lipids. So this is a synthesizable lipid space. All the lipids are synthesizable by this 4-component chemistry, combinatorial chemistry. You pick up a component in one category, and it's like you have Lego components and make out of them art, and in one compound, and they are combined together in this formulation. So by this way, we have a large space, but all the model can propose candidates, out of this huge large lipid space. In each of the round, we only pick up around 190 lipids. Within 10 iterations, I will show in later stage, we actually select a very nice candidates. So that is only a very small portion of the whole space. The full workflow of the 10 iterations working this way. This is like we have a balanced exploitation strategy for the active learning. So in one plate of the experiment, so I think this, sorry, the font is a little bit small. Yeah, this one. So this blue one is the more traditionally active learning strategy. So it selects the most uncertain candidates after each iteration. But we also have another one because we want to synthesize the candidates as fast as possible and finding the most promising ones. We have this exploitation plot, which testing the most promising candidates proposed by the models after each iteration. So it's a combination of the candidates. We see so this one shows the experiment measured transfection efficiency after each iteration in each row. And we saw this consistently increasing performance among iterations. We use so after 10 iterations, we look at the most promising ones. We found that LUMI Lab successfully found normal bromination strategy. So it does not only found promising targets, but actually a novel strategy of new family of lipids. So we have a lipids that are containing this new bromine atoms, and all the top 6 in vitro tested lipids fall actually into this category. We test them also in vivo to deliver the CRISPR-Cas9 gene editor into mouse lung. It actually give us the best in class efficacy, and in comparison with other strong baselines like this MC3 and SM102 lipid nanoparticles. So this SM102 is an actual LNP that used in the COVID-19 vaccine. We also test this newly discovered lipid nanoparticle ionizable lipids head-to-head in the way that we remove the bromine lipids. We remove the bromine atom. You can see just the one atom difference. You see this 1.8-fold change of their efficiency. So it does verify that the improved performance, improved efficacy is achieved by our new strategy. So the LUMI Lab's result provides us empirical conclusion that this guided exploration, this data collection steps, and they combined with the optimization of the model, and it can be an efficient way to at least finding some local minima in your very large tested space. So a recent work of Evo Pro, I just want to cite this work, also shows some similar results, but on different directions. They use that to also achieve the successful stories in editing and optimizing gene editors and other proteins. So the workflow, I think this workflow is an example, and we can use that to we can borrow this workflow for perturbational training for building next generation single cell foundation models as well. To illustrate, we can have a initialization stage. It's like the stage we train a base model. It's like what I already talked about in previous slides. We use the public domain data, combine all the different modalities. We can train a multitask pre-trained model. But we do recognize, the limitation of the data we currently have in large scale. Although they are in large scale, but a lot of perturbational data, they are one gene perturbation, and they can be tested in cell lines rather than in actual human cells or tissues. But given this initialization, we can have a base model, and then we can have a similar workflow like the loops of laps in the loop. The model proposes the candidates, we test that quickly in vitro, and then feedback to the model to do some fine-tuning and then propose a GUIN. The nice part about that, I just give an example here, but the really nice part I want to highlight that although this lap in the loop can work rather slow, but it can work in a focused way. And you can also have an outer loop. It's like your different applications. You can imagine you have a series of applications that you actually collected the biased data. It's not IID data, but actually model-biased data, model-inspired data in different applications. And the data, things they are in small scale, most of the time we can test it in more interesting cell lines and in more interesting gene pools. So this is actually accumulating the most valuable data for the applications you really care about. Combined with the large data we already have, I envision this can be actually a good way for the model a pre-trained model and your data co-evolve together. In the short term, just the last sentence is that in the short term, I think this is a more promising way or more feasible way that we have a model and data co-evolve together rather than just envisioning a single large model that solves everything just in silico. So to summarize, the talks this talk today is first I introduce the lessons learned from scGPT and the importance of the training and the inference alignment, and briefly mentioned the trend about building future virtual cell models. And very lastly, using the example of LUMI Lab, I think the outer loop of active learning is a very good way to co-evolve the data and model together. Thank you very much. I think I already saw some of the questions in the chat, and
Yeah.
also like to express my deepest appreciation to all my collaborators for their contributions. Thank you, and willing to take any questions.
Thanks a lot. And we do have a few different questions, which I will very quickly, ask. One is related to the scGPT. Was asking, are you like having any component in the loss explicitly to deal with the batch effects, or is it removing it by itself by basically having a large amount of data? So that's question number one. And the second part is asking if you mostly use the CLS token when you did the KNN, or also use the contextual gene embedding as well.
Yeah, the first part about the batch effects. Yeah, we did we have two things related to that. For the pre-training itself, most of our work is done during the data pre-processing. We have the value binning, which I want to summarize. It's just like every time it's like the quantile normalization. So every time you look at one cell and doing in a very extreme way, you just relatively rank all the expressions and then binning them from high expression to very low expression. So in a sense, every time the input to the model is just showing whether the gene is relatively high expression for the cell or low expression. The absolute magnitude of the gene expression is then removed. We found that this can be a very extreme way, in the sense a lot of the signals are missing, are removed. But it does remove a lot of batch effects. Even just after this pre-processing, if we have 2 or 3 data sets and we process the data and then do the PCA on the data, and for some data sets, usually you have huge batch effects. Now after the same PCA, we do saw fewer batch effects, in the sense we saw some clusters of cell embeddings, and this was a intuitive evidence we had. I would say this was the most hand-made strategy we use in the pre-training, and this might contribute, I would say maybe 50 or 60% of why the pre-training can work, even on huge batch effects of hundreds of different studies. The other part is just the self-supervised pre-training. I personally think is a good way that is more resistant to a batch effect as well. And the second part of my answer is that actually during the fine-tuning, a lot of fine-tuning tests, we had some strategies, additional strategy, but that it was more commonly used or introduced earlier, such as one of the interesting one is that we do a thing called gradient reverse gradient backpropagation, which is sort of like adversarial training. So you can have the batch label, but you're training the model in a way to unlearn the batch label rather than optimize to predicting it. That was a strategy introduced very early, I think 10 years ago, for the general domain adaptation field, but we borrowed that for the fine-tuning stage. We didn't have that in pre-training because after the first part of the answer, we saw the pre-training itself already works well. Yeah. Sorry, I forgot the second part of the question.
It was related to whether you only use the CLS or also contextualized gene embeddings when you talked about clusters of cells or doing KNNs.
Oh, for KNNs, for any cell-related task, we only use the cell embedding, which is a CLS token.
Okay. And then the second question is around the second part of the talk, which is the LUMI model. Basically, what is the model predicting, or I assume how the loss is exactly?
Yeah, it's once again, it's similar to what we talked about before about the misalignment between pre-training and inference. So the inference itself is quite easy. You just have a model, and the model can generate this time a molecule embedding, some vectors for different molecules, and use that. You have some classifiers appended after that, and then the classifier is say continual pre-trained or fine-tuned in your active learning loop to predict a score. In our case, is a transpaction potency of the RNP. That's the score is the output. But during the pre-training, the output is different and trained differently. But basically, there's two stream pre-training. One is like contrastive learning. The model is so you did some augmentation. We did some augmentation over the. This is trying to represent the distorted coordinates. Also, some of the atoms tabs are masked, and so first you use the model to reconstruct this noise input, and also you do contrastive learning in this using this already augmented input. Yeah.
Thank you.
If I can step in there. Well, thanks so much for the talk, Haotian. So you're suggesting that during pre-training, these different tasks pull the model in different directions, and that combining them should help capture this broader view of the biological landscape. But at first glance, one might expect that the masked modeling is close to perturbation modeling, right? So in both cases, the model must infer missing information from the context, right? So I'm wondering if there is a more principled way to map where these different tasks align and where they fundamentally differ, so that you know what tasks are complementary to include during pre-training and what have overlapping type of information. Is that something you've thought about?
Yeah, this is a really great question. I mean, I thought about this, but honestly speaking, I don't have an answer for that. And I think if we can, so currently, I think myself and also other people working on this direction, we are doing this trial by error and trial and error way. Is like we just try different tasks and we see whether they can work well together or not. But it would be super nice if we can have a not gas, but if we can have some small scale tests that can tell us whether the task we wanted to add, whether they can work together or not beforehand, that would be super nice. Currently, we don't have that. I only have the strategy to, you just try on a smaller model, a fewer data set, and see, using this kind of tasks, this kind of tests as your way to justify whether the task could work together. So in theory, I do think the reason I pick up I think I have a slide is maybe this one small visualization. I do think the perturbation actually is another dimension. You if you only learn, no matter what you learn currently in single cell RNA-seq data via the self-supervised learning, you don't know the T+1 stage of the cell. You are now prompting the model to do anything related to time, to future time points. So I do think this is a missing piece, and the overlap where between the tasks are actually small in this way. Yeah, that is one part. Another interesting thing, I always think this case by case, and in theory, I didn't find a unified way. I just want to share another example we did find interesting is a cell type annotation. So during the pre-training of scGPT, we didn't use any human annotation. And that was because the annotation was noisy. Imagine you have 400 different studies. But later on, we found that if you actually train on a smaller data set, but they are homogenized and curated with high quality of annotation, for which I mean the Tableau sapiens, that data set, 50 million of high quality human cells. And sorry, I don't think it's 50 million. It's I think it's 500,000. But anyway, we found that actually can give the model more capability of recognizing cell type. So even though you have the self-supervised pre-training, some additional information right from the annotation, like from the human expert view, expert knowledge, can still help, at least in our experiments. Yeah. Those are some examples I just had for how these tasks may work well together, or maybe they may not.
Yep. It makes perfect sense. I mean, I've been thinking for quite some time now, what is the right way of determining what these models are learning? And like you can think of doing linear probing and stuff like that. But I guess if you combine multiple tasks and you see that the performance improves, I guess that's an indirect way of saying that, yes, these tasks are quite different from each other. And so maybe including them all together in doing pre-training might help learn better biology, I guess.
And one last question we also had in the Q&A, which is in more applied direction. How much do we think have we come so far in understanding biology? For example, can we use these models to simulate how diseases develop and how to cure diseases in like in different disease areas?
It's also a great, but honestly speaking, open question. So I think we are in the early stage. If I may say a timeline in terms of analogy to the actual GPT, like the GPT-1 to GPT-5, I think maybe we are somewhere between GPT-1 and GPT-2, and maybe around GPT-2. So it's in the stage that we can use a model. It sort of tell you something, but the in silico does not replace the in vitro in any way. Maybe 10% of the in vitro test, but not a significant way. So, but the positive answer for that is that what personally gives me a huge of confidence is that I did saw a lot of users, if they fine-tune the model on a high quality private data they have, or some specific question they have, if it's a focused high quality data for a focused question, focused application, they find this can be a very good model, and that can predict or using some words from the question to predict some cell state, or for our users, usually they care about perturbations. We did have some successful stories. They recapitulate some cell states that matches very well in their experimental data. I think the missing piece is that how we can merge from a fine-tuning of the pre-trained model to something with less fine-tuning to be more few shot, and even more to a day to be zero shot. I think this is ongoing, and this is definitely happening. Yeah.
Thank you so much. I think that concludes most of the questions. Are there any other questions on your end, Eugen?
I think we're good.
Yeah. Okay, and then I wanted to thanks again from both participants and our presenter, and yeah, we are gonna stop it here. Thanks, everyone.
Thanks so much.
Thank you.
Mhm.